Welcome back, dear students and friends. We are running this chapter number 2. It is sexual reproduction in angiosperm flowering plant. In this chapter, up till now we have finished two basic events, pre-fertilization and the first fertilization. Now we have to move ahead with the post-fertilization events. And in the post-fertilization events, we will discuss today two events. First is the formation of endosperm and the second is the formation of embryo. You know that now we are starting with pre-fertilization. Post-fertilization. In previously, we have seen that in the fertilization, in angiosperm, the fertilization occurs two times. As a result of first fertilization, it leads to a formation of a zygote, a 2N zygote which is going to be formed. And the second one, it leads to a formation of triploid primary endosperm nucleus. These are the results of fertilization in angiosperm. First product is zygote, which is diploid. The second product which is going to be formed is a PEN, primary endosperm nucleus. It is a triploid. Now we have to see, this zygote, it leads to a development into an embryo. And this endosperm, primary endosperm nucleus, that converts into an endosperm. So here, both the processes occurs by mitosis. This process as well as this one, the division that occurs by a mitosis process. So the embryo which is going to be formed is a diploid because in mitosis the chromosome number remains same. And here the endosperm or the cells of endosperms are triploid because the chromosome number remains the same because the same process, mitosis. So that occurs in a post-fertilization event. So today we have to see how the endosperm is going to be formed from primary endosperm nucleus and how the embryo is going to be formed from zygote. Apart from these events, means formation of embryo and formation of endosperm, the ovule after fertilization, ovule gets converted into a seed, and ovary after fertilization that converts into a fruit. So we also have to see these events, and they are also post-fertilization events. Ovule converts into a seed and ovary converts into a fruit. So ultimately these four events occur after fertilization in flowering plant. Zygote converts to an embryo, primary endosperm nucleus converts to an endosperm, ovule converts into a seed, and ovary converts into a fruit. In this session, we will discuss these two processes, how the zygote converts to embryo and how primary endosperm nucleus converts to an endosperm. Okay students? So let's start. One of the first event in a post-fertilization is endosperm. So you know that endosperm is a post-fertilization event. This event occurs after fertilization. So that's why we call it as post-fertilization event. Okay, endosperm formation. This endosperm formation starts before the embryo formation. Okay, the endosperm formation starts before the embryo formation. Why it is going to happen? Because the cells which are produced in an endosperm or the endosperm tissues, they are filled with a reserve food material. This reserve food material has to be supplied to a developing embryo. And so to get the assured nutrition, first endosperm starts to develop. Once somewhat endosperm has been developed, afterwards embryo formation starts. Okay, so this is endosperm formation. How it is formed? The primary endosperm nucleus gets converted to an endosperm tissues or the cells of endosperm which are going to be formed. As I have already mentioned that here the cells or the chromosomes which are present in primary endosperm nucleus is a triploid. So the endospermal tissue also has a triploid number of chromosomes. Okay, that is a 3n number of chromosomes or their cells are triploid in nature. How it occurs? This primary endosperm nucleus, it contains a nucleus. This nucleus undergo a repetitive mitotic division. See, remember, it's a repetitive. So many nuclei are going to be formed inside the endosperm. Okay. Once this nuclei are going to be formed, these are called as a free nuclei which is going to be formed. Afterward, there is a formation of cell wall around it. And as it is a plant cell, the cell wall formation occurs from periphery and then it extends towards the center. Okay. So this type of endosperm formation occurs in the angiosperm or the flowering plant. I will give a very good example. Like you might have seen a coconut. Coconut contains a water, coconut water. What is this coconut water? The coconut water is free nuclear endosperm. They are the nuclei of endosperm tissues. And this coconut water is surrounded by a white kernel. Okay, this white kernel is an example of a cellular endosperm. So in an endosperm formation process, it occurs in a two-step. First, the nucleus which is present in the primary endosperm nucleus, this starts to divide repetitively. Multiple time division occurs, and so that's why the multiple nuclei are going to be formed. Okay, and then later on cytoplasm divides, and then cellular endosperm is going to be formed. Okay, these endosperm cells, they are filled with reserve food material, and this reserve food material which is present in it, that is supplied to a developing embryo. Okay. But during the endosperm formation, now you know that the cells of endosperms, they are filled with a reserve food material, and that food is supplied to a developing embryo. In some plants, this developing embryo is utilized during embryo formation. The food which is present in endosperm, that is utilized by the embryo, fully utilized, no food remains. But like in the case of examples, you can say a groundnut, pea, bean. Okay. So in this case, the embryo utilizes the food which is present in endosperm fully. Like for example, groundnut, pea and bean. In some cases, the food which is present or reserve food which is present in the endosperm, that is not utilized fully by the developing embryo. It remains. And the remaining food that is utilized during the seed germination, like which happens in the case of castor, coconut, where the food is not fully utilized, means partially utilized food by embryo. Okay, the remaining food that is utilized during the seed germination process. Okay. So the ultimate purpose of this endosperm is to provide a nutrition to a developing embryo. In some seed, that nutrition is fully utilized before the maturation, or at the maturation the food remains nil. Like for example, in case of groundnut, pea and bean. And in some seeds, the food remains partially utilized. Okay, like in case of castor, coconut, the remaining food that is utilized during the seed germination. Okay. So that is all about the endosperm formation. So in short, if I want to say then how the endosperm forms, the endosperm forms from the primary endosperm nucleus by repetitive mitotic division. Initially only nucleus gets divided and it forms a many nuclei. Like for an example, coconut water, it's nothing, it's just a free nuclei. Okay. Then after there is a formation of a cell. Okay, so many cells are going to be formed, what we call it as endosperm cells or endosperm tissues. Okay, in a coconut, the white kernel which is going to be formed around the water, that is called as a cellular endosperm. Okay, and you have to remember the examples. The examples of a seed in which the endosperm is fully utilized. Which are the example? Groundnut, pea and bean. And the endosperm in which the food is not fully utilized, or the seed in which the endosperm food is not fully utilized, castor and coconut seed. Okay, so that's all about the endosperm formation. Okay. Now we will start a formation of embryo. A second process after fertilization. Okay, so the another event in a post-fertilization is formation of embryo, and that is very important step. Okay, it is a post-fertilization event. Embryo formation, and you know that embryo is formed from zygote. this zygote, which is diploid in nature, that converts into a embryo by a mitotic division. Hence, the embryo is also a diploid in nature, means it contains 2n number of chromosomes. Okay, by which process they convert? By a process called as mitosis. Okay, let's first discuss how this is going to be formed. You know that this is embryo sac. Embryo sac, usually it contains 7 cells. You know that which are the 7 cells. Total 8 cells. 8 cell converts to a 7 cell. 3 at the chalazial end, what we call it as antipodal cells. 3 antipodal cells at the chalazial. This is micropylar end. And it contains 1 egg cell and 2 synergy cells. And in the center there is a presence of polar nuclei. Okay. Now you know that this polar nuclei fuses with a male gamete. 2 male gametes are there, and they fertilize 2 times. So that's why what we call as double fertilization. This polar nuclei gets converted into a PEN. Okay, by fusion of polar nuclei and male gamete. The 1 egg cell, this 1 egg cell that converts to a zygote when it is fuses with a male gamete. Clear? So now what we have? Zygote at micropylar end and the PEN that is in the center. The development of endosperm starts from PEN, primary endosperm nucleus. And now we have to see from zygote the embryo formation starts. Okay? So where this zygote is present? The zygote is present at the micropylar end. You have to remember where this zygote is present. The zygote is present at the micropylar end. Okay? So I just enlarge this cell, the zygote cell. Okay? And then we will go ahead. Suppose assume that this is a zygote 12. I am just enlarging this cell. Okay? A zygote. Now how the embryo formation process occurs, that we have to understand. So for easy understanding, I am enlarging a view. Okay, this whole is embryo cell from primary endosperm nucleus, endosperm have been developed, what we have discussed. Now from this zygote, how the embryo formation process that we have to see. Okay, now you know that this zygote, where it is present? This zygote is present at the micropylar end. So whatever the process occurs, that occurs at the micropylar end. Okay, and so what process occurs? Initially, this zygote, it divides transversely. And it forms one large cell and one small cell. Okay, what it forms? It forms a one large cell and the one small cell. The large cell which is going to be formed, that is called as basal cell. And the small cell which is going to be formed, this is called as apical cell. Okay, basal cell is formed towards a micropyle and the apical cell is formed towards a chalaza. This is towards chalaza and this is towards a micropyle. Towards chalaza, this is chalaza, towards chalaza. Okay, upper cell. And this is the basal cell which is towards a micropyle. Clear? Two unequal division occurs, not equal. So one large cell and one small cell. So it's a transverse division. After that, what happens? Afterwards, we have a condition like this. One basal cell, one apical cell. Basal cell is towards a micropyle, the apical cell is towards a chalaza. This basal cell divides transversely. Clear? And the apical cell, it divides vertically. So now, how many basal cells we have? We have 2 basal cells. And we have how many apical cells? We have 2 apical cells. Clear? I will repeat. The basal cell, this is present, that divides transversely. So 2 basal cells are produced. And the apical cell, it divides vertically. So how many apical cells are produced? 2 apical cells are produced. These 2 are apical cells which are going to be produced. Now it forms a 4 cell structure. 1, 2, 3, 4. This 4 cell structure, it's called as proembryo. It's called as proembryo. Okay, this is the first step in embryo formation. Zygote converts to a proembryo. Okay. How the zygote converts to a proembryo? The cell of a zygote, it divides transversely and unequal cell division occurs. So that's why one large cell and one small cell is going to be formed. Large cell which is present towards a micropyle is called as basal cell. The small cell which is towards the chalaza is called as apical cell. Now this basal cell divides transversely and the apical cell divides vertically, and it forms a group of 4 cells, and this group of 4 cells is called as proembryo. But for this division, it needs energy, it needs a food. So how it gets a food? The food which is present in the endosperm, as I have already told in the previous session or previous topic, that the food is already present in the endosperm. That food is utilized by this zygote or developing embryo. Okay, and then it forms proembryo. So that's why the embryo formation process, this embryo formation process starts once the small amount of endosperm is going to be formed to get assured nutrition. Okay, so endosperm બની જશે પછી જ embryo formation process ચાલુ થશે, અથવા થોડોક endosperm બની ગયો હશે પછી જ embryo formation process ચાલુ થશે, because the embryo has to take a nutrition from endosperm. Okay, now here the first step we have finished. Zygote converts to a proembryo. Now what happens to this proembryo? You know that proembryo contains 2 basal cell and 2 apical cell. From this 2 basal cell and 2 apical cell, the basal cell towards a micropyle, this is a 2 basal cell, one is towards a micropyle, one is towards a chalaza. The basal cell towards a micropyle remains as such. Okay, and the basal cell which is towards a chalaza, it starts to divide and it forms a filament like structure which consists of group of 20 to 25 cell structure. How many cells? I have drawn only few because of lack of space, but there are a group of 20 to 25 cell structure is going to be formed. This is called as suspensor. What it is called as? Suspensor. This is a basal cell. Okay, one of the basal cell remains as such. The basal cell towards a micropyle, that remains as such. Now the basal cell towards the chalaza, it continuously divides and form a group of 20 to 25 cell structure, or 20 to 25 cell structure, they are arranged like a filament. What we call it as suspensor. And above that, what is present? There are presence of a apical cell. You already know it. These are 2 apical cells. These 2. Okay. Now the cell of a suspensor which remains in contact with apical cell. This is a cell of suspensor that remains in a contact with a apical cell, and this is called as hypophysis. The large cell of a suspensor which remains in a contact with a apical cell, it's called as hypophysis. Clear? So I can repeat. In a proembryo, it's a group of 4 cells. From these 4 cells, there are 2 basal cells, 2 apical cells. From 2 basal cells, the cell towards a micropyle, this is a cell towards a micropyle, remains as such. But the cell towards the chalaza, it continuously divides and form a group of 20 to 25 cell structure called as suspensor. It's like a filament. One of the cell of suspensor that remains in contact with apical cell, what we call it as hypophysis. Okay. During the meantime, when this process continues, during the meantime, this apical cell also divides and it forms 4 cells. 4 cell again divides, it forms 8 cells. Now see 8 cells, 1, 2, 3, 4, 5, 6, 7, 8. This 8 cell again divides and it forms 16 cells. Now we will count, there are the 16 cells. These 16 cell structures, they are grouped into 2 parts. The upper 2 part, it is called as chalaza octant means contain of 8 cells, and the next one is, it's called as micropylar octant, because the upper one they are present towards the chalaza, the lower one they are present towards the micropyle. So it's called as gelazal octant, or the lower one is called as micropylar octant. When this process occurs, during the meantime the same process is going to be occur. And ultimately, what do you say? There is a formation of the shape. Shape is going to be changed, and it's going to be changed like this. Heart shape. These are the 16 cells. This is a suspensor. And this is a big basal cell which has not divided. So there is a formation of heart-shaped globular embryo. Okay, what it is formed? It forms heart-shaped globular embryo. Now we are discussing with respect to a dicotyledonous seed. You know that dicotyledonous seed, that means it contains how many seeds? How many cotyledons? It contains 2 cotyledons. So what it leads? It leads to a formation of 2 cotyledons. So these are 2 cotyledons. It is dicotyledons, and this is called as embryonic axis. So now this heart-shaped globular embryo is converted into such structure in case of dicotyledonous seed. So what the dicotyledonous seed contains? It contains 2 cotyledon and 1 embryonic axis. What it contains? 1 embryonic axis. The portion of embryonic axis above this cotyledon, that is called as epicotyl. What it is called as? Epicotyl. And the portion of embryonic axis below the cotyledon is called as hypocotyl. What it is called as? Hypocotyl. Remember, the dicot seed contains 2 cotyledon and 1 embryonic axis. The portion of embryonic axis above this cotyledon is called as epicotyl. The portion of embryonic axis below the cotyledon is called as hypocotyl. Okay. Now this epicotyl, it contains plumule, and that develops into a shoot system. And this hypocotyl, it contains radicle and from that root, cap and root apex is going to be formed, or you can say a root system is going to be developed from it. Clear? That happens in the case of dicot seed, dicotyledonous seed. So how the process occurs? The heart-shaped globular embryo converts into a such structure. This is a full of cells. Like this, number of cells are present, lots of cells, they continue to divide, it's full of cells. Clear? But for easy understanding, I am drawing a structure like this so you can easily remember this structure. Okay, so it contains 2 cotyledon and the 1 embryonic axis. And the portion of embryonic axis above the cotyledon is called as epicotyl. The portion of embryonic axis below the cotyledon is called as hypocotyl. The epicotyl develops into a plumule develops into a shoot system. Hypocotyl, that has a radicle, the radicle develops into a root system. Thus, from seed, the shoot system and the root system develops in case of dicotyledonous seed. Now in monocot seed, in dicot seed there is presence of 2 cotyledon. But in monocot seed there is only a presence of, this is a difference. In monocot seed there is only a 1 cotyledon and it has embryonic axis. Other thing is same. The portion above it is called as epicotyl, the portion below it is called as hypocotyl. Epicotyl that develops into a plumule and the hypocotyl that has a radicle. Okay, but there is a one difference, or what are the changes in the term? In case of monocot seed, there is only a 1 cotyledon and that cotyledon can be called as, I am writing over here, it's called as scutellum. What it is called as? Scutellum. the cotyledon of monocot cell. What is another name? It's called as scutellum, a large shield-shaped cotyledon found in monocot cell. It's called as scutellum. We have also learned it in the 11th science. But what is the difference? This epicotyle, or you can say a plumule, that is surrounded by a covering. That covering is not absorbed into a dicot cell. The covering of plumule in that is called as coleoptile. What is called as covering? Covering of plumule, and covering of radicle is called as coleorhiza. That is found into a monocot cell. Remember, the covering of the plumule is called as coleoptile, covering of radicle is called as coleorhiza, and its cotyledon can also be called as scutellum. Okay, so that's all about a formation of embryo. I again repeat, it is very important process, can be asked in full marks, 4 marks, 5 marks. Okay, usually it's going to be asked. So how the process is going to happen? Zygote, where the zygote is formed? The zygote is formed towards a micropylar end. Okay, zygote is formed towards a micropylar end. This is the structure of embryo cell. That's for easy understanding. Doesn't require in diagram. But I just want to show that here the zygote is formed towards a micropylar end. I have just enlarged this zygote over here. It contains a 2 end. This number of chromosomes. This zygote divides transversely, form of one large cell, one small cell. Large cell is called as basal cell. Small cell is called as apical cell. Now this basal cell divides transversely. Apical cell divides vertically. Form of 4 cell structure called as proembryo. This is a proembryo which is going to be formed. Okay. Now this proembryo contains 2 basal cell and 2 apical cell. From 2 basal cell, the basal cell towards a micropyle, this is a basal cell towards a micropyle, remain as such. But the basal cell towards a chalaza, it continue to divide and form a 20 to 25 cell structure called as suspensor. One of the cell of suspensor that remains in contact with this apical cell, and this apical cell, the cell which remains in contact with cell of suspensor which remains in contact with apical cell, what we call it as hypophysis. During the meantime, this apical cell also starts to divide. So initially we have how many cells? 2. It divides, 2 cell becomes a 4 cell, 4 cell becomes a 8 cell, 8 cell becomes a 16 cell. But remember, each division occurs right angle to a previous divisions. So ultimately how many cells we have formed? 16 cells are formed. These 16 cells are divided into 2 groups containing 8 cells each. 16 cells divided into 2 groups. One group contains 8, another group contains 8. The one group that contains 8, that is called as chalaza octet, which is towards the chalaza. The other group which contains 8 cell called as micropylar octet, towards a micropyle. 8 and 8, total 16 cell. And it acquires which shape? Heart shape. This heart shape, this is the second step. It's formed, acquire a heart shape. So this is called as heart shaped globular embryo. And it continue to grow up, and many cells are going to be formed. In dicot cell, it possesses 2 and so that's why there is a development of cotyledon and embryonic axis. In dicot cell, there is a presence of 2 cotyledon and 1 embryonic axis. The portion of embryonic axis above cotyledon is called as epicotyle. The portion of embryonic axis below cotyledon is called as hypocotyle. Epicotyle develops or contains a plumule and that develops into shoot system. Hypocotyle that develops into a radicle and that form a root cap and a root apex. This is about a dicot cell. In a monocot cell, there is only a presence of 1 cotyledon and 1 embryonic axis. The portion above the cotyledon is called as epicotyle, as like dicot cell. The portion of embryonic axis below it is called as hypocotyle. But here you can see the cotyledon which is present in monocot cell, it can be referred as scutellum. Okay. Here the plumule is covered by a covering. This covering is called as coleoptile, and the radicle that is covered by covering that's called as coleorhiza. That is the difference. Okay, and thus the embryo formation occurs in a angiosperm or flowering plant. So today we have discussed 2 processes. One is endosperm formation and second is embryo formation. Remember, the embryo formation occurs after the formation of endosperm because the formation of embryo requires a nutrition, and that nutrition has been provided by endosperm. So the first endosperm formation starts and then embryo formation starts. Okay, so that's all about it. The remaining, how the seed is going to be formed, how the fruit is going to be formed. You know that ovule converts into a seed and ovary converts into a fruit. So which are the components of a seed, how the seed is formed and how the fruit is formed from ovary, that we will see in the next session. Okay, so till then keep watching, keep learning, like and subscribe. Thank you for watching.